guys, Her Majesty took away my fear I had in chemistry and biology. You have to join the easy ones. My name is Victor Modia, a student in Italy. Anywhere you are, you can join us. Join the easy one. Be a subscriber. Bye.
So we are done with the filtrates according to the instruction. The next is the residue. According to them, the residue say with the help of spatula. We should now add the residue in a boiling tube and add about 5 cm of H2SO4. Okay. According to the question, they say we should add about 5 ml of dilute H2SO4.
Hi, my name is Jordan and welcome to Her Majesty Easy World channel. This is the best channel you can get on YouTube. So make sure you like, comment and subscribe.
Welcome to the Easy World of Science. Today in our salt analysis to prepare you for the forthcoming examination, I will be analyzing a mixture of two salts which is actually copper 2 oxide over here is a black powder just like manganese oxide. Then with zinc tetraoxosulphate 6, I will mix the two salts and then analyze them according to the questions here. So, and then the question that we will use for this particular analysis, they will tell this question is telling us that C is a mixture of two inorganic compounds. Carry out the following analytical procedure on C or G. Either record your observations and the conclusions from the result of each procedure. Identify any gas or gases evolved. Detailed account on the experimental procedure is not required. So 2AI says put all of G in a beaker and add about 10 ml of, that's 10 milliliter of distilled water, stir well, filter, keep both the filtrate and the residue, test the filtrate with litmus, divide the filtrate into 3 equal parts. Then the question II says, to the first portion of the filtrate, add sodium hydroxide in drops until in excess. To the second portion of the filtrate, add aqueous ammonia in drops plus excess. To the third portion, they ask us to add barium chloride followed by excess dilute HCl. That's aqueous barium chloride followed by excess dilute HCl. Then the B part of it says to the resulting, the B part of it says with help of spatula put all the residue in a boiling tube and add about 5 cm of dilute H2SO4, that is dilute tetraoxosulphate 6 acid, and warm gently. The following question now says to the resulting solution from BI add aqueous ammonia in drops till excess. So this is the question guide we need in this analysis, and that's what I'll be using to analyze. So what I need in this analysis, those things needed are I need my sodium hydroxide aqueous, then dilute H2SO4, barium chloride solution, then aqueous ammonia, ammonium hydroxide, then hydrochloric acid is also needed. So these are the reagents that I will be required to have for the analysis of this mixture of two salts. Then other things I might be need for heating is my Bunsen burner, and here is the wash bottle. Then we have the filter, because we have mixture of soluble and insoluble salt. We have filter paper here. Then normally the petri dish is here. Then we have here is the litmus paper, blue and red. These are the necessary things. You might also have your glass rod and the spatula. Then we have here the test tubes. The test tubes are, first of all, we are required to mix these two samples. Okay, here is the spatula. I'll get the samples. I'm going to get equal mixtures of the two. Let me start with this. Spatula. Okay, this is the final thing we have in the mixture of the two mixtures of the salts. Remember, this is the unknown sample. So we get our question guide and see the instruction first. They say put all of G in a beaker and add about 10 ml of distilled water. So this is my beaker. This sample is too much. I won't be analyzing the whole because if it is in exam, one student cannot get all this. It's too much. I'll just scoop small. Just a student will get only 2 spatula full or 1 spatula the maximum. So this is enough. Okay. Let's put this aside. This is the water. They said about 10 ml. Remember, it's about 10 ml. So accuracy in the volume is not necessary, but this test tube is nearly 10-20 ml. So I'll take half. Just get what I can divide into 2 portions well. Okay. With the help of glass rod, I'll stir well. save time, keep it here.
Hi, my name is Cheryl.
And I'm Jennifer.
Guess what? There's a channel called Her Majesty Easy World Science.
And do you know the best part?
It
makes science so easy. Wow.
It makes science easy, simplified, and very, very fun.
Guess what? Rocket science ain't rocket science anymore.
It's now ABC.
Like if you did science your entire life.
Another thing about Her Majesty Easy World Science channel is that he makes available laboratory equipment and reagents.
Guess the best part? If chemistry has been hard for you, he does tutorials.
And another thing is, when you order for these things, they are high quality and they are also cheap and affordable for anybody.
If you want to order, just look at the number below the screen.
And don't forget to subscribe and hit the little notification button down below.
Don't forget to share, of course, obviously there's love in sharing.
Thank you! We'll see you there.
So in this case, you have carried out the first instruction, the first test given. So as you can see now, we have two things. This is a colorless filtrate or white filtrate, that's colorless. Then the residue is black. So that would be your observation. Your observation is a colorless filtrate and a black residue. So we take the next step, which says that we have to divide these filtrates, according to them of course, into 3 equal parts. So here I have the test tubes. Any test tube you will use, you wash very well to avoid contamination of your results. So I'm going to use these 3. Okay. 3 equal parts doesn't necessarily mean you must be accurate. Just into 3 portions. We have the 2 here, our stock solution. Okay. Let me bring these out. These are my 3 portions. Our guideline question says, to the first portion of the filtrate, add sodium hydroxide in drops till excess. So I'm going to add sodium hydroxide, which is here, in drops till excess. So in my report, under the column for test, I'll write first portion of filtrate plus NaOH aqueous in drops plus excess. So this is my dropper. I will use it to add the drops. Of course, in drops, I'm seeing white gelatinous precipitate in drops. White gelatinous, it is sticky to the walls of the test tube. So you can watch other videos here and test for cations for you to see gelatinous, meaning like a gel sticking to the walls. It's not like a free powder. So in drops we have white gelatinous precipitate. Then in excess you don't need a dropper, just pour. So in excess it gives us precipitate dissolves. The white gelatinous precipitate dissolves to give a clear solution. So our observation is white gelatinous precipitate formed in drops which dissolve in excess. So what's our inference? Anytime you add sodium hydroxide to an unknown sample and it gives you a white gelatinous precipitate which is soluble in excess, you are advised to suspect zinc or aluminium. These are the 2 things that will give us gelatinous precipitate with aqueous sodium hydroxide. But for the sake of staying on the safer side, you can add lead, of course. So lead also forms white precipitate with sodium hydroxide, but not gelatinous. So you can write down in your inference zinc, aluminum, or lead is suspected. So there are 3 ions that can form white precipitate with aqueous sodium hydroxide in drops and will dissolve when you add excess of the aqueous sodium hydroxide. But I say the difference between the two is that aluminum and zinc are gelatinous, they are sticky, while that of lead is powdery. Now, why will these 3 ions behave similar in the presence of sodium hydroxide in drops and in excess? Because both are amphoteric. The hydroxides they form are amphoteric hydroxides, which means in the presence of excess sodium hydroxide, they will react to form a complex salt. So that is the reason why zinc, aluminum, and lead behave similar with sodium hydroxide, because both are amphoteric metals, or either their hydroxides are amphoteric, meaning they have both basic and acidic properties. Now, using our question guideline, I'm done with this. I'll keep it here. This is the remaining 2 samples. The other one says, to the second portion of filtrate, add aqueous ammonia in drops until excess. So I will be using aqueous ammonia. You can see it as ammonium hydroxide. So this is the second portion. So it's advisable you also wash the filter or the dropper rather. Let me use, you can use your hand and add the drops, just small. Okay. Formed white gelatinous precipitate. When I added it, there wasn't. Then in excess, it dissolves again to be very, very clear. So white gelatinous precipitate formed in drops, which dissolves in excess to give us a clear solution, just like we saw in sodium hydroxide. So what is my observation here? I said white gelatinous precipitate formed in drops, which dissolved in excess. Maybe I repeat this experiment, this particular stage, so that you can see the gelatinous nature of the precipitate. So these are stock solution. And I want to add the drops using the dropper. Yeah. So that's the precipitate, white gelatinous. So then it is sticky to the walls. Then in excess, the gelatinous precipitate will disappear and become clear. See the solution, the precipitate has dissolved to give us a clear solution. Remember, this is aqueous ammonia. So we observe the same thing when we use sodium hydroxide and aqueous ammonia. And when I use sodium hydroxide, I told you that you suspect aluminum, lead, or zinc. But mostly, if it is gelatinous, it should be zinc or aluminum. But when the same result is observed with aqueous ammonia, you confirm zinc ion. Because zinc ion, that's zinc 2 ion, is the only cation that will form a white gelatinous precipitate which is soluble in aqueous ammonia. The other 2, meaning remaining aluminum and lead, will not dissolve in ammonia, but they will still form precipitates like so that of they will form precipitate with aqueous ammonia, but they will not dissolve in excess. The reason why zinc is the only one that will dissolve among the amphoteric metals, meaning zinc, aluminum, and lead, why will zinc dissolve in ammonia among the 3? Because zinc is a transition element and forms a complex ion with ammonia. Remember, ammonia is a ligand. In other words, it's an electron pair donor and is a good Lewis base. So it donates a pair of electrons, which now forms a coordinate covalent bond with the zinc metal. Remember the properties of the transition metals, the ability to form complex ions. So our inference, I say, since it's aqueous ammonia, is zinc 2 ion, confirmed. So we now go to the next one, the third portion. Our question guide says that we should add barium chloride to the third portion, aqueous, followed by dilute HCl. So this is barium chloride aqueous. Here they are not talking about in drops or in excess, just add. So let's add. Okay, see, there is formation of white powdery precipitate with barium chloride. Then in chemistry, anytime you add barium chloride to solution of unknown sample and it forms white powdery precipitate like this, you suspect three anions which are SO42 minus, SO32 minus, or CO32 minus. That is tetraoxosulphate 6 ion, or trioxosulphate 4 ion, or triazocarbonate 4 ion. The reason is this barium is a kind of double decomposition. This barium here will go in here and react with either SO4 to form barium sulphate, because barium sulphate is insoluble. If CO3 is here, the barium will react with the CO3 to form barium carbonate, which is also insoluble. If SO3 is here, the barium will react with SO3 by exchange of radical to form barium trioxosulphate 4, which is also insoluble. So the idea is trying to check if any of this is there, then you precipitate it out. Now, I've told you in your inference, you suspect SO42 minus, SO32 minus, or CO32 minus if you add barium chloride to any solution of unknown sample and it gives a white powdery precipitate. Then, which reaction will tell you the one that is present? The next thing, according to the instruction, says barium chloride followed by excess dilute HCl. So it is the HCl that will tell us which among these three is present, or which among these three, 2 out of the 3 might be present. Now, the next is to add the HCl. This hydrochloric acid will now determine, will group these 3 into 2. Meaning, if this precipitate dissolves, it means SO32 minus or CO32 minus, because barium carbonate and barium sulphide, that's BaSO3 and BaCO3, are all soluble in acids. But if it is SO42 minus, this precipitate will persist in HCl because barium sulphate is insoluble in acid, as it is also insoluble in water. So, let's check. The precipitate is just dispersed, not that it is dissolving. If you look at here, it is still the way you shape this well. See, it is still there. So the precipitate persists, is not dissolving, it remained milky as it was before. That means what is present here is tetraoxosulphate 6 ion. So in your inference, when you add barium chloride, write SO42 minus, CO32 minus, or SO32 minus, suspected, then immediately it persists. In dilute acid, you write SO42 minus, confirmed. So this has confirmed that the unknown sample, the G or the C I'm analyzing, contains sulphate 6 ion. So we are done with the filtrate according to the instruction. The next is the residue. According to them, the residue, say with the help of spatula, we should now add the residue in a boiling tube and add about 5 cm of H2SO4. Okay, this is my residue. Okay, open the filter paper. This is the residue. So here is my spatula. Here is the boiling tube. Boiling tube is just a bigger test tube. So I will scoop. This is my spatula. I'm going to introduce this into the boiling tube. Can use this. Okay. At least there is. We have gotten it into this boiling tube. That's the black residue is there. According to the question, they say we should add about 5 ml of dilute H2X4 to the residue. You can use this to run down the whole thing. So you're adding and then warm gently. See, even before warming, it's slightly dissolving. So the next step is to warm gently. See, on warming is dissolving to form a blue solution. And whenever blue comes into analysis, you think of only one cation, which is copper 2. So it's showing that our residue is probably containing copper 2 ion. So definitely the residue has dissolved. So your inference should be black residue dissolves to form a blue solution. In your inference, copper 2 ion likely. So they now say to the resulting solution that we should add aqueous ammonia in drops until excess. So I want to get accurate result. In this case, use your discretion because the aqueous ammonia coming in is a base and the acid here, so it is also there. So there might be a chance that the ammonia will be used up by the acid. That the ammonia will go first, neutralizing the excess acid. So to get an accurate something or more precise something, just collect small sample over here and continue your experiment. So we are adding the aqueous ammonia in drops. In drops there was a light blue precipitate which is happening. It has disappeared because there is excess acid using up the remaining ammonia for neutralization. So you see the blue color coming out very well. This is light blue gelatinous precipitate. So let me discard a bit so that I'll have space to add excess. Okay, then in excess, you see, dissolves to form a deep blue solution. So in your inference, your observation is with aqueous ammonia in drops forms light blue gelatinous precipitate, as you can see here at the base. Then in excess, the light blue precipitate dissolves to form a deep blue solution, and that confirms copper 2 ion. So from this analysis, you have seen how to test for copper 2 ion and also how to test for zinc ion, the confirmed zinc ion, and also how to confirm tetraoxosulphate 6 ion. Remember, Sa Majesty Easy World Science channel cannot do without you. So endeavor to share my videos, expand the science community, and you can also look for your own private classes for instructing in practical chemistry, in biology, and equally some of the medical related courses. You can check other videos on analysis on this channel. I'm happy having someone like you. I cannot do without you. Much love from me to you. Remain majestic. Don't forget to subscribe and share. Like, ask your questions there in the comment box. Bye for now.
I love you. Thank you for watching.
And